Hello everybody. This video is a review session about DNA, and specifically we're going to talk about what DNA is, what its structure is, how it gets replicated in the process of sexual reproduction for a single-celled organism or growth for multicellular organisms. And then we'll talk about the process of gene expression via transcription and translation. And finally, we'll talk a little bit about the process of mutation. So let's get started here. DNA's role is that it contains genetic information. And we can think of the genetic information contained in genes, which are specific locations on chromosomes. And chromosomes themselves are long linear strands of DNA in eukaryotes, which you can see right here. And in prokaryotes, this should show a circular strand, but for illustrative purposes, they made it linear just to make it parallel, but it's really a circular strand of DNA. At any rate, the genes occur at certain locations along the chromosomes, and they contain information. And an overview of that process, of how that information gets expressed, what we call gene expression, is that the genes get transcribed into various RNA molecules. In other words, the complementary RNA gets made from the DNA. And there are many types of RNA, and we learn about more and more of them every decade. We're going to talk about three types and then a fourth general type. The first type is known as mRNA, messenger RNA, and that carries the information to make proteins. So first, the info in the mRNA is turned into a polypeptide, and polypeptide is just a chain of amino acids, by a ribosome, and that process is called translation. So you can see that here. We have transcription occurring here and translation occurring outside the nucleus on a ribosome. The polypeptide then is folded into shape so that it can become a functioning protein. And the processing of these polypeptides often occurs in the Golgi, although not always. And we should say right now that most of transcription creates messenger RNA, but there are other types that we've learned about. So we've also learned about rRNA, or ribosomal RNA, which is used to make the structure of the ribosome. So this organelle here is mainly made up of RNA. And we've also learned about tRNA, or transfer RNA, which carries the amino acids from the cytoplasm to the ribosome and makes sure the right amino acid is put in the right place during the process of translation. And then we also saw other RNAs that we didn't specify that are involved in the processing of the pre-mRNA that is made directly by transcription into the final or ready to translate mRNA. And that's carried out by RNAs. And that process does not occur in prokaryotes, only in eukaryotes. So let's now go to what does the DNA molecule actually look like? Well, the building block molecule for DNA is what is called a nucleotide. And the nucleotide itself is composed of three other molecules. It's composed of a sugar, which is here shown in blue. And in the case of DNA, that sugar is deoxyribose. In the case of RNA, it is ribose. It consists of a phosphate, a PO4 molecule that gets attached to the sugar, and it consists of a nitrogenous base. Now, you will notice what I mean by that is these bases have a lot of nitrogen, or several nitrogen atoms in them. It is important to note the structure of the nucleotide. The carbons in the sugar can be numbered 1, 2, 3, 4, and 5. And in the nucleotide, the base is attached to the number 1 carbon and the phosphate to the number 5 carbon. That's very important for the structure of DNA. What we are going to see, what we know about DNA is that there are 4 types of nucleotides. 2 of them have 2 rings, and 2 of them have a single ring. So we have A, C, G, and T, which stand for adenine, 2-ringed, cytosine, 1-ringed, guanine, 2-ringed, and thymine, 1-ringed. Now, in DNA, the nucleotides are chained together into the nucleotides are strung together into a chain, and these chains are then wrapped around each other in the famous double helix. But first I want to look at that chain. The backbone of the DNA is composed of this chain made of sugars and phosphates, and it's important that you look at this and see how this is done. Here is an adenine nucleotide. You can tell it is because here's the number 1 carbon attached to the base and the 5 carbons attached to the phosphate. And the way it attaches to the thymine above it in this picture is through the phosphate, which bonds to the empty 3 carbon in the thymine, and that's the backbone. Similarly, this cytosine, its phosphate, bonds to the number 3 carbon, or binds to the number 3 carbon in adenine. And this guanine nucleotide binds to the number 3 carbon in cytosine. The bases, as a result, dangle off to the side, and that's going to become important. Now, when the 2 chains are wrapped around each other, there are 2 important rules. And I think you know these important rules, and they're illustrated here. Here's a picture of the double helix where you can see that it's not just a flat molecule as shown in this representation, but it's a three-dimensional helical structure looking much like a spiral staircase where the base pairs are the rungs of the stairs. So let's talk about the 2 rules. The 2 rules are these: that G always pairs opposite C and T always pairs opposite A. So the strands are said to be complementary because if we know what is on one strand, then we automatically know what is on the other strand. And that turns out to be very important information in the replication of DNA, as you already know. Now, the other rule is that one of the things I didn't mention back here is that the chain has a 5' end, in other words, a phosphate that dangles off one end that's attached to the number 5 carbon, and a 3' end where there's an empty 3rd carbon that doesn't have a nucleotide attached to it. So the chain has a directionality, and that is shown in this illustration here, where you can see the 3' end points down in this illustration, and the 3' end on the opposite chain points up. As a result, we say that the 2 chains are anti-parallel. They run in opposite directions. Now, you already know that this causes great problems, or better said, it causes complications when DNA needs to be replicated. The fact that they run in opposite directions means that we have issues during replication. Why this happens is a great question of chemistry and evolutionary biology. My guess is it happens. The reason for the anti-parallel evolution is it creates a more stable molecule because these hydrogen bonds better line up and can stabilize the molecule in a much better fashion. Okay, so that's it for the structure of DNA. This is a good time to stop the video and make sure that you've got everything I've said here in your notes. And it's a good time to figure out questions to ask me. We're going to pick it up next. So now what we're going to look at is the replication of DNA. And the first question to ask is, why does DNA need to be replicated? Right? And so the answer to that is, whenever a cell needs to reproduce itself, which in the case of single-celled organisms means it's reproduction itself, but in the case of a multicellular organism like yourself, it's regeneration of damage, maintenance of parts, or growth requires cell division. So whenever cell division has to happen, each daughter cell, and that's the terminology we use, daughter cell, needs to have a complete copy of the DNA. Okay, so each daughter cell needs a complete copy of the DNA. That's the reason for replication. So how does DNA replication work? Well, the overview is that the original DNA gets pulled apart, and then the complementary side is built by enzymes using the information on the old strand to form the complementary strand via the rule, if it's a G, you put a C opposite. If it's a T, you put an A opposite. If it's a C, you put a G opposite, etc., etc. The process of replication is carried out by 5 enzymes. These enzymes, and here's the picture of that, have the names helicase, here depicted in green, which unwinds the strand, primase, which puts down an RNA primer, and we'll talk about why that is in a second. No, let's do it now. Puts down an RNA primer because the next molecule, DNA polymerase, cannot just copy on naked DNA. It needs a 3' end to work. So the primase puts down a primer in RNA, which is not stable. DNA polymerase 3 then comes along, and it puts down DNA until it bumps into the next primer. Now, at that point, it can't work anymore. It drops off, and a new enzyme called DNA polymerase 1 is able to remove the RNA primer and put DNA in its place. But the problem is it cannot bind. It can bind onto a 3' end over here, but it can't make a bond with this 5' end of the piece of new DNA. That's left to the final enzyme in the process, DNA ligase. And ligation means to tie, so that's where that name comes from. So we have the 5 enzymes: helicase, primase, DNA polymerase 3, DNA polymerase 1, DNA ligase. They each have a job to do. Now, remember that each strand serves as a template. And so in theory, that's a very easy rule to follow. But in practice, because the DNA is opening up, each strand is copied in a different fashion. So let's look at that. We call one of these strands the leading strand and the other the lagging strand. On the leading strand, the DNA is copied continuously because the primer was laid down back here and the copying is going into the opening, and therefore it never needs to stop. That's the leading strand. It can be done continuously, as you can see up here. And it will be continuous until the DNA polymerase runs into a piece of primer when it runs into the next one of these bubbles, which are termed the origin of replication. So you can imagine that there is another origin of replication out here somewhere, and this is just going to keep copying until it runs into that piece of primer that looks like this. On the lagging strand, which is the other strand, the copying is done away from the origin of replication. And so what that means is new DNA is constantly being opened up, and there is nothing there. So primers have to be laid down every once in a while. Now, how long that is, I don't know. The video we saw implies it's maybe 100 or 200 base pairs gets looped out before the primer is laid down. And so the DNA is copied in fragments here. We call them Okazaki fragments. You can look up how to spell that if you need to. And so once again, what that you can see here, that process. Here is the oldest piece of DNA that was new DNA that was laid down. This now, number 2, is the second oldest, and you can see it still has some of its primer. The second oldest piece is now being joined to the oldest piece by ligase. The third oldest piece is currently being finished. The primer is being removed by DNA polymerase. And so that will soon create a gap here. The fourth oldest piece is just now being made by DNA polymerase, which is going to stop when it hits this piece of primer. And the fifth oldest piece hasn't been made yet because the primer is just being laid down for it. So I hope that clarifies for you this complexity of the leading and the lagging strand. One way to think about it is the leading strand is copied into the opening of the DNA, and the lagging strand is copied away from the opening of the DNA. Now we want to jump to how DNA, the information, now we want to jump to how the genetic information is actually made real. And that's the process of transcription and translation that we reviewed earlier. What I want to do now is actually go through transcription and translation in more detail. This is a good time to stop the video and make sure that you understand replication and write down your questions about replication. Okay, so now we're going to look at transcription and translation in more detail. So, transcription, of course, is the making of RNA from DNA. And it's made by a gene called RNA polymerase. If the gene codes for a functional RNA, an RNA that is not a messenger RNA, in other words, then that RNA diffuses throughout the rest of the cell and does its things. We've learned nothing about them except what they do, what ribosomal RNA does. We know they get taken somehow to the nucleolus, they get shaped there into the ribosomal subunits, and then those subunits get transported into the cytoplasm where they are put together. If the gene codes for a protein, then the mRNA is first processed in the nucleus. And that itself is carried out by other RNAs, as I said earlier. And what happens? What happens there is, and I'll go down and show you this, is that what got originally transcribed was a series of what are called coding regions, the exons, and that stands for expressed regions, and non-coding regions called the introns, or intervening regions, or intervening sequences, as well as promoters and tails that are not going to be put into the final RNA. So in the processing, all this extra DNA, sometimes referred to as junk DNA, but in this case it has a very useful function, keeps the exons separated, is excised or cut out, and then the exons are spliced back together into a functional messenger RNA. So that's what happened here. We took the pre-mRNA and turned it into a functional mRNA. Now, there are two other things that happen. I didn't talk much about it, but there's a cap put on one end of the mRNA, and the other end has a poly A tail, and that's simply a lot of adenine nucleotides put on the end. And the purpose of that seems to be that as soon as this mRNA gets into the cytoplasm, there are enzymes that want to recycle, better said, whose job it is to recycle the nucleotides, the mRNA back into its nucleotide building block molecules. So this poly A tail protects the message of the mRNA until it can be transcribed. And then once it's transcribed, it can be recycled. So then the mRNA, once it's made, diffuses and finds a ribosome. Those ribosomes are either free-floating or on the endoplasmic reticulum, and then the polypeptide is made. How that gets sorted out, how the mRNA knows to go where, I don't know. There must be some sort of molecular signaling that's doing that. Now, that's transcription. Let's take a look at translation. The first thing we want to look at from translation, so here we see a picture. Here's the DNA template. Here is the transcript. And in translation, the messenger RNA is read as a triplet code, or the nucleotides are read in sequences of 3. So in this case, UGG, we call it a codon, and it codes for the amino acid tryptophan. UUU codes for the amino acid phenylalanine, GGC for glycine. This is the famous genetic code, and on any test, you'll get a copy of this genetic code that you can use to figure out what's going on. So, now, let's take a look at the genetic code for a second. Let's go back to the genetic code. The interesting thing to note about this code is that all 64 three-letter combinations code for something. 61 code for amino acids, and 3 are stops, which mean they tell the ribosome to stop making the polypeptide. So that's important. Now, it turns out that methionine, the codon AUG, is the first codon in almost every protein. And that's an interesting question as to why that happens. You'll study that, what's known about that in an advanced course. And so the first amino acid in most proteins is methionine, although sometimes that can actually get excised and cut out, from what I understand. So now the process of translation, the ribosome latches onto a piece of mRNA and then begins to read it. Now, the ribosome itself, which I've often likened to a giant brioche hamburger bun, has 3 sites. It has what's called the attachment. So here's the ribosome, the brown unit. Here is the mRNA moving through it. And here are the 3 sites. The first site is called the attachment site, or the A site. It's where the tRNA is going to attach. The second site is called the polypeptide site, because when the new tRNA attaches, the growing polypeptide is held by a transfer RNA that is now situated in this middle site. And then finally, so that's the P site. And then finally, we have the ejection site, right, which is the site where the spent tRNA, the tRNA that no longer has its amino acid, and here you can see it has its amino acid, is ejected. So those letters spell out, if you read them, EPA, Environmental Protection Agency. I don't know if that helps you remember it. It helps me remember it. If you have a backwards mind, it spells out ape. And what are humans but just wonderfully evolved apes? Now, the idea here is that the tRNA has an anticodon that is the complement of the mRNA codon. So here we see GGC, and we see here that the anticodon CCG is coming in to match with this, right? And so the tRNA, the ribosome will only accept the tRNA that has the right anticodon, and in that case, it'll bring the right amino acid in. If we look at the actual cycle of translation, we have an empty A site, and now the tRNA that is the complement of the next codon comes in and is temporarily attached there. Then energy, here represented by this molecule called GTP, you can just remember it as cellular energy, is used to transfer to bond the new amino acid to the polypeptide chain. And then once it's bonded, more energy has to be used to move the whole thing to the left, at which point the old tRNA is now ejected, and is shown here that can go back out into the cytoplasm and get an amino acid reattached and then come back and participate in translation again. So that is translation. And I think this is a good time to pause the video and see if you have any questions, to finish your notes on translation, and to see if you have any questions about translation that you need to ask me in a review session. Finally, we're going to get on to mutations. So let's do that. Mutations are the result of the incorrect copying of DNA during replication. We reviewed replication earlier. And the mutations we've learned about, although there are other types, can either be a substitution of one base by another, or an insertion or deletion of one or more bases. Right? Now, mutations may change one or more of the amino acids that are put into a protein. Right? That's important to realize. And mutations that don't change the amino acid sequence or that don't affect the functioning of the protein are called silent mutations, because they basically have no effect even if they've created a change in the DNA. Most mutations are either silent, which means they don't affect the protein, or they are lethal. They affect it so badly that the organism doesn't develop. And that's another reason why we may not notice them, because the embryo aborts spontaneously. Occasionally, mutations must actually improve the functioning of the protein, and in that case, those individuals may be subject to positive selection, where they actually leave more offspring to subsequent generations as a result of that mutation. It may enhance some aspect of their survival or reproductive abilities, and then they'd be subject to either natural or sexual selection. So, let's look at these mutations. But before we do, one last thing that I want to say to you is that mutations, base pair substitutions that still code for the amino acid, but not the same amino acid, or still code for an amino acid, but not the same amino acid, are called missense mutations because they've changed the sense of the protein. But when the changed base pair results in an early stop codon, this is called nonsense because that now becomes, almost always, it's a non-functional protein. So what is made doesn't make any sense to the cell. So that's how these work. And here's a picture of this. Here you can see that the original DNA had a C and now a U is here. Well, the codon GGC and GGU both produce glycine. So in this case, the mutation is silent and has no effect on the amino acid sequence, and therefore it won't have any effect on the protein. Now here's an example of missense, where instead of the G here, an A got copied in there, and now the amino acid is changed from glycine to serine. Once again, remember, you do not need to know the names of these amino acids, nor do you need to memorize the genetic code. You'll be given that always. Now, in this case, it's unknown whether the serine here instead of glycine will affect the protein. Because it's at the end of the protein, it's possible that it won't affect the protein. And so it may be a silent mutation as well. But that is a missense mutation. Finally, we could have in this codon where it should be AAG, a U placed in the first base. That makes, in the fourth base. And that makes this codon UAG. That means everything after methionine is not copied. And so this creates nonsense because this protein is likely not to be functional, missing 3 amino acids. Although there must be cases where the last 3 amino acids being lopped off may not make a difference. So that's substitution reactions. Let's look at insertion-deletion mutations. In an insertion-deletion, it's just what it sounds like. An extra or many extra nucleotides are put in, or some are taken out, one to many. So look what happens here. What happens is when you put a new mutation in, and here you can see a U is put in here where it should be UUG. It now makes the codon UUU and then GGC. Oh no, it's taking them out. I'm sorry, this is an elimination. And so what happened in this case is now instead of it being UUU, it became UUG. That changes it from phenylalanine to lysine. The next codon, which should be GGC, is now GCU. That changes it to alanine. So now this is why it's called frame shift, because every group of 3 down the line is read incorrectly. It would only be coincidence or lucky coincidence if the same amino acid was put where it's supposed to be. So that's frame shift that causes extensive missense, right? And that's a frame shift mutation, very likely to cause a non-functioning protein. Here is an example where frame shift, whereas an extra U is put in, causes an immediate stop codon to be put in, and we don't make the rest of the protein. So those are 2 examples of frame shift. Now, frame shift when it is in multiples of, insertion deletion when they are in multiples of 3 are less likely to harm the protein. In this case, AAG has been taken out, which means that the lysine is missing, but methionine, phenylalanine, and glycine are still there like they're supposed to be. And this protein may be able to continue to function, or it may not. It could be a lethal mutation. So any insertion or deletion of 3, of a multiple of 3, will have an effect on the protein, but its effect is likely to be less than any insertion deletion that is not a multiple of 3. I hope that makes sense to you. Because when it's a multiple of 3, there is not frame shift. That's the answer to that. So now's a good time to write down, finish your mutation notes. Make sure you understand it. Write down any questions you have for me. But that's it for our review as well. So make sure you go through this and that you bring questions to me before we get to our test. And I'll see you soon. And if you have a chance, when you're not freaked out for studying, go outside, take a look at things in the natural world, and ask biological questions about them, and bring those back to class. See you soon.